All right, hi everyone. This is our lecture on chapter 2, which is titled Genetics, Genomics, and Epigenomics. Okay, so essentially our genetics chapter. There is some basic genetics and basic biology in here, and so if there are additional questions, just on anything, just make sure you ask me to clarify if necessary. All right. So, in terms of the learning outcomes that are presented here in this chapter, we can read through them, I guess, here. So, first one, describe the impact of genetics, genomics, and epigenomics on personalized healthcare and explain the concepts related to them. Okay. And then, like I said, there's some basic genetics here. So, identify the components of the genetic code and the organization of genes on chromosomes, understand the differences between the human genome and epigenome and the mechanisms by which epigenetic modifications occur and affect gene expression. Okay, explain the function and sequence of events involving gene replication during mitosis and meiosis. Very basic, so I'm not really expecting you guys to know every single detail there. This is really just an overview. There's a lot more detail that we can talk about. I mean, we could probably spend, a month. I mean, you probably could spend almost a whole semester just talking about really the nitty gritty details that are involved in a lot of these processes that are mentioned here. So we really want to know big picture. Okay, so just keep that in mind as I'm going through the lecture. And we'll again mention transcription and translation. Why isn't this changing? Okay. Oh, sorry. Yeah, I was going to say, I know I missed one. We're on the second slide here, right? So 2.5, compare single nucleotide polymorphisms and the various types of gene mutations. So we'll talk about different types of gene mutations. We'll then talk about characteristics and patterns of inheritance of just genetic disorders, right? So talking about chromosomal abnormalities as well. Differentiate the mechanisms responsible for phenotypic variations in human disease, a little bit about that, but again, some of these we're not going to get into much detail about it. Understand the role of genetic and epigenomic factors in cancer. Again, I'm not sure that we understand the complete role, but we'll touch on that. We'll talk a little bit about some tests that could be used in a clinical situation, like karyotyping, FITC, which is fluorescence in situ hybridization. Oh, that's fish. Sorry. There's another there's another dye that's called FITC. Anyway, fish you sometimes see this abbreviated as. So we'll talk a little bit about that. We'll talk about some genetic based therapies. And then just talk a little bit, sort of recap and talk a little bit kind of where we're at in terms of how some of these things might impact personalized healthcare. I think we hear a lot about personalized healthcare and personalized medicine. Obviously, that all involves genetics. Okay. All right. So for that first learning outcome, describe the impact of genetics, genomics, and epigenomics on personalized healthcare and explain the concepts related to them. So this is more the kind of just the introduction. What do these terms mean? What concepts are related to these? Okay. So just like I said, kind of just like an overview and introduction to the chapter. So if we talk about a gene, right? We're talking about genetics, genes. A gene is just a segment of DNA that codes for a specific protein. Okay, so you can have a gene, right, that encodes for any specific protein that your body produces. So I don't know, you can think of, I guess, any sort of an enzyme or something like that, right? Or think of a peptide hormone or something like that, right? Or I don't know, I always think about kind of the opioid system, right? Or the opiate system. If you think about things like endorphin, right? We've probably heard of endorphins before, like runner's high, and given this increase in endorphins. Those are, again, they're going to be produced by a specific gene. There's a lot of more complexity surrounding that, but you can kind of think about it that way. Okay. If we talk about genetics, we're talking about the study of individual genes. Okay. So, you can think about, inheritance of these individual genes, right? And then if you think about disorders that might be associated with this, right? So you can have single gene effects, right, that can lead to disease, but then you can have also a little bit more larger scale where you might have a duplication of a chromosome, an entire chromosome or something like that. Okay, and we'll talk about that, and that can lead to disease as well. All right, so again, just to kind of decipher or delineate between these three terms here. Genomics, study of structure and function and analysis of the whole genome, right? So we think about the genome being all the genes that exist and sort of how that's related to disease, if we want to think about it in the realm of pathophysiology. If we think about epigenetics, right? External modification of the DNA that affect gene expression. So now this is not changes in the DNA sequence, or we're not really talking about the sequence of the DNA at all. We're talking about things that modify the DNA in some way and then can affect the expression of genes, right? So it affects, it's not going to cause a change in gene sequence, but can have just a similar effect as a change in gene sequence. And we'll talk more about that. Epigenomics, study of chemical compounds that instruct the genome where and when genes are expressed within the cell. So now you're bringing in those molecules that are going to have the epigenetic effects. Okay, so you can kind of see, and again, I mean, sort of take those definitions with, I guess, with a grain of salt, but it's just sort of meant to kind of distinguish between the three. Okay, to give you a little bit more clarification there. All right. I think I mentioned this Healthy People 2020 in the first chapter, and basically, you know, this is a whole bunch of government agencies that came together to kind of put forth these goals and these sort of I guess criteria and standards that all sort of healthcare personnel should be considering, right? And so actually one of the goals that was added was to improve health and prevent harm through valid and useful genomic tools in clinical and public health practices. So meant to sort of, right, to promote the use of genetic tools via genetic testing, genetic treatments, and what have you. Okay, so you can kind of read through this. I'm not going to obviously read through it. You can read through it a little bit on your own just to kind of get an idea about this, right? So it says here, it tells you about the objectives here, right, related to genomics target cancer, right? And the two objectives of Healthy People 2020 related to genomics target cancer. I guess that's kind of slightly a typo there. And then they're also to increase the proportion of women with a family history of breast cancer, right, to receive genetic testing. So again, the Healthy People is about, I think, kind of telling healthcare providers what are some important things that you should be conveying to your patients. And one of them are this, right? So if you have a family history of cancer, you should be going to get genetic testing, right? And so it's to make people aware of that. If you take a look again, these are one of these types of figures you're going to see as you move through the textbook, as we move through every chapter. And it's kind of, remember we had that table in chapter 1 with all these concepts and sub-concepts. Well, this is again kind of a offshoot of that table, right? So if we particularly look at genetics here and genomics in the center, we know that genetics can affect things like cellular metabolism, right? Regulation of cells, right? Perfusion, how well, blood is going to move through your blood vessels, reproduction, right? All of these sort of main topics here. And then you can actually then see what types of diseases are then associated with that main function, right? So if we talk about genetics impacting metabolism, that can ultimately maybe lead to something like diabetes. There's other examples we can add in here too. If we think about genomics impacting cellular regulation, we probably think about cancers related to that, right? So if there's some sort of genetic change that causes cells to divide more often than they should, then that is going to lead to cancer. If we think about genetics impacting perfusion, going to lead to things like heart disease, right, and stroke. Okay, and so hopefully we kind of understand what this figure is showing you. Again, see, this is sort of illustrating the broad impact of genetics on health, right? Or health and or the opposite of that, disease. Okay, all right. So let's start off by identifying some components of the genetic code and organization of genes on chromosomes. Again, this could be a review. I mean, you probably touch on this in an anatomy and physiology course. You definitely talk about this in a general biology course. So I don't, it kind of depends on the background that you come from. Okay, so first thing, if we're going to talk about genes or genetics, we got to talk about DNA, right? And so DNA stands for deoxyribonucleic acid. The deoxy Ribopart is the sugar backbone, and I'll show you a picture of it in a second. I'll make it a little bit more clear. So we know this is the building block of human genetic material and contains thousands of genes, okay? And when we think about a gene, again, a gene is going to code for a protein, says it provides instructions for body function, right? The genes themselves do not provide. The gene's role is to provide that information to make a protein. The proteins are then the functional molecules of the cell. Okay, so that's what that is getting at here. If we talk about gene composition, right? Genes or DNA are made up of a sequence of nucleotides, right? We talk about those 4 bases. So when you think about a nucleotide is composed of a phosphate, a deoxyribose, which is the sugar, right? And one of 4 bases: adenine, guanine, cytosine, and thymine. And so we talk about DNA, or we talk about gene sequence, we're talking about sequence of A, G, C, or T. Okay, so that's all DNA is made up of these 4 different bases. Okay, and obviously then creates so much diversity and information just from a sequence of these bases. Okay, so if we look at a picture here, what this is showing you, if we talk about nucleotides, right, they're composed of, like we said, a phosphate, the sugar, right, because this is essentially the backbone of the DNA, and then one of these 4 bases. So we talk about purine, right? Purines or purine nucleotides, adenine or purine bases, adenine and guanine are purines. And you can see that their structure, they have 2 rings. If we talk about pyridamines, cytosine and thymine, they are pyridamines. And we always have a purine pairing with a pyridamine if we talk about, right, DNA, and we talk about complementary base pairing, okay? And more specifically, we always have A pairing with T, right, and G pairing with C, okay? And so, like I said, DNA structure, where I'll show you a picture again and clarify what I just sort of said, but you have 2 separate strands of these nucleotides. And again, A pairs with T, G pairs with C, okay? And so those bases that we talked about are the nucleotides, right? The A and the T are joined together with hydrogen bonds, okay? You have this double helix that forms, and I already mentioned the purine and the pyridamines, okay? So I probably already mentioned all of this ahead of this slide, so sorry, but again, this will all be clarified again in the picture. So we talk about purine bases, adenine and guanine, pyridamine bases, cytosine and thymine, and we talk about complementary base pairing. T always pairs with A. If there's a T there and some reason there's not an A, that's a mutation, right? That's not how it's supposed to be. And there are a lot of mechanisms that can repair things like that. And we'll talk about mutations a little bit later on in the lecture. Okay. So I apologize for this being small. Unfortunately, I don't know. I feel like the figure could be a little bit bigger, but it does the job, right? So again, here's showing you the structure of the DNA. So the purple is that sugar, essentially phosphate backbone, right? The green is the little yellow guys are the phosphates, and these are the sugars. And then you can see the bases attached, right? And so we can see here, so again, here's one strand, again, forming this double helix, and here's the other strand. Complementary base pairing, G pairing with C. What this is just showing you in the middle here are hydrogen bonds. Okay, look, guys, this is not I'm not going to be asking you really like specifics, like how many hydrogen bonds are formed between G and C. That's not what it's about. However, we should be able to recognize that this is the structure of DNA. We should know that C pairs with G and that A pairs with T. We should know that the backbone of DNA is made up of deoxyribose, the sugar, and a phosphates attached to it. Okay? So again, this is the basic structure of DNA, and also showing you the base pairing as well. Okay, so we already said this, right? We said it has this double helix composition. When we think about though, and so that's really zooming in on the sort of specific structure of DNA, but when we think about how DNA is packaged or how it exists sort of inside our cells, inside the nuclei, it has to be packaged in a certain way, right? And it's organized in chromosomes. We've heard of chromosomes before. The way that DNA gets packaged essentially into chromosomes is due to it associating with proteins called histones. So the DNA can wrap around these histones, and that's what's able to compact it into this chromosome form. So if you think of a chromosome right, is DNA and proteins. Okay, it's just a different level of organization. So there's DNA there in those chromosomes, right? But there's also proteins that help organize it in that way. Okay, so that's what it says here. Each chromosome contains many genes, right? Also contains proteins, these histones, and also other proteins as well. Okay, so that's what this is showing you here, right? We saw that picture before where we were specifically looking at the DNA, right? And that double helix structure. Well, like I told you, when we think about it inside the cell and inside the nucleus, it's packaged in the form of chromosomes, okay? And so all the chromosomes are here are proteins and DNA that essentially that DNA is wound around those proteins, okay? So that's what this is showing you here. A little thing here, it says each cell contains 46 chromosomes. So we should know that there are 46 human chromosomes, right? 2 pairs of 23. There's 2 meters of DNA. So that's how that DNA, that 2 meters of DNA, think about that. That's a lot of DNA, is wound and packaged around these proteins and able to fit inside this tiny little nucleus, okay? This is just giving you some numbers here to really kind of think about. 3 billion DNA subunits, 25,000 genes code for proteins that perform all life functions. Okay. All right. So again, if we talk about those chromosomes, they're located inside the nucleus like we just saw. Okay. Most of your body cells contain 46 chromosomes, 2 pairs of 23. 23 chromosomes that came from your father and 23 chromosomes that came from your mother, okay? There's an exception to this. Red blood cells actually don't contain nuclei, okay? And so they don't contain chromosomes. They're really just membrane, essentially. Okay. Egg and sperm cells, again, exception. Egg and sperm cells contain 23 chromosomes. Your sex cells only have 23 chromosomes because, again, if you think about when the sperm cell fertilizes the egg, you then join those two pairs of 23 and end up with the 46 chromosomes. Okay, so that's what this is saying. Chromosome pairs formed at conception when that sperm fertilizes the egg. Okay, again, 1 chromosome inherited from each parent. That's what I was just telling you. You have 23 pairs, right? 1 from your mother and 1 from your father. 1 paternal and 1 maternal. Okay, again, I mentioned this. Your sex chromosomes, your sex cells only contain 1 pair of 23. Okay, so if we talk about those sex, and then we think about our chromosomes in general, the 23rd pair of your chromosomes, right, are the sex chromosomes. Okay, so, and it's going to differ between males and females, right? So females have 2 X chromosomes, males have an X chromosome and a Y chromosome. Okay. It's mentioning here a bar body, right? Inactivated X chromosome in each female cell. So there is, because the X chromosome contains more genes than the Y, it's actually necessary to inactivate one of those X chromosomes as you sort of move through in those female cells. Okay, don't worry. Again, these are things that like we could talk about in a lot of detail. We're not going to. So to be honest, probably not going to ask much about bar body, but we could talk about like this process, right? How that happens, but that is beyond the scope of this class. Okay. One thing we really should know the definition of is alleles, right? So alleles are alternative forms of an individual gene, right? So when we think about, again, we have 2 pairs of chromosomes, 1 maternal and 1 paternal. And if we look at those chromosomes and we look at specific genes on those chromosomes, right? So this is showing you this. Gene for kidney function, right? Gene for kidney function here. If you talk about your chromosome from your mother, your maternal, and your chromosome from your father, on each one of those chromosomes, you're going to have this gene for kidney function. Now, you have alternative forms though. So I don't know, the form that you get from your mother may be different from the one that you get from your father. Okay, so that's what we mean by alleles. They are alternative forms of that same gene. Okay, also giving an example here for skin pigment. Okay, all right. So again, make sure we understand what allele means, right? We can and we're going to talk a little bit about some genetic diseases, but think about this, like, one of these being some sort of disease allele. So if we think about, I don't know, cystic fibrosis or something like that, where you have a diseased allele, right, versus a normal allele. Okay. And so, depending on what type of disease you're talking about, it can lead to disease or not. And so we'll talk about that, but that alternative forms. Okay. All right. Also, worth mentioning is that there is DNA in the mitochondria. So mitochondria have their own circular DNA. That's always going to be inherited from the mother, right? Maternally inherited, because again, you have the egg cell, right, which has all these other organelles in there, including mitochondria. The sperm cell basically just fertilizes that egg and kind of gives its DNA. So essentially, but so there's no mitochondria coming from the sperm cell, put it that way. So that's why it's always maternally inherited. Okay. But there are several important metabolic genes that exist in that mitochondrial DNA. And so you can have mutations in the mitochondrial DNA that then can be passed on from your, right, maternally, right? They're always going to come from your mother, and they can actually lead to disorders, diseases. Okay. And that's beyond the scope of this. If you're interested in something like that, look it up, right? Read a little bit more about it. But there are genes that are particularly part of the mitochondrial DNA, okay, and passed on that way, again, only through the mother. Okay. So moving on to the next learning outcome, right? Understanding the differences between the human genome and the epigenome and the mechanisms by which epigenetic modifications occur. So we'll talk a little bit about epigenetics here. And kind of what we were talking about before was really kind of basic genetics, right? So now we're kind of taking a step up here to talk about the genome, genomics, and and epigenomics a little bit here, okay? So again, if we talk about the genome, it is the complete set of DNA within an organism, okay? Again, we said this before, almost all human cells contain the entire genome, okay? I'm sure we probably have heard of like the Human Genome Project, right? So we have sequenced the entire human genome. We've also sequenced the genome of many other animals that are used in research as well, which has been super helpful in understanding the impact of genetics on disease, okay? I already gave you this example before. Genome size will vary greatly from one organism to another. Okay, we've found that out as well. And if we talk about the human genome, and I think I mentioned this a little bit later on again, but 99.9% of the human genome is identical between individuals. Okay, 99.9%. So that's saying there's about a 0.1% difference between individuals. Okay. So if we talk about size, right, 3.2 billion base pairs in the human genome, about 22,000 genes. Okay? Again, it says known genes make up only about 2% of the genome. The remaining 98% of the genome is not involved in encoding proteins. So some other numbers that are probably a little bit surprising. Okay? So this means 98% of the genome is not involved in encoding proteins. Okay? Most of these are involved in maintaining chromosome structure and also important in regulating gene expression in some way. And so I'll just tell you that, like, I don't, at one point, we used to kind of call the DNA that did not code for proteins as junk DNA. Obviously, it's not junk DNA if it's 98% of the genome. So by sequencing that human genome, I think, made us understand how important these non-coding regions of DNA are. And then there's been lots and lots of studies studies involving those regions of DNA, okay? And those regions of DNA obviously can impact disease. All right. So I already kind of mentioned all this to the Human Genome Project, sort of ahead of that, so I apologize. But again, if we think about genomics, we're talking about studying the entire genome. We're not talking about studying individual genes here, okay? And you can look at, mechanisms of health and disease within the scope of the entire genome, okay? So again, it's not the study of individual gene function, it's larger scale, all right? So again, I mentioned this, the Human Genome Project sequenced, right, identified and mapped the entire human genome, okay? And I already mentioned this too, we sequenced model organisms used in research. Why is that important? Because we use model organisms, right, in research to model disease, to model human disease. So we probably need to know how similar the genome is, right? Or if we have similar genes that are implicated in human disease, also in our animal models. Okay, so super, super important that we were able to compare this. Okay, so before we had the human genome sequence, didn't have that ability. Okay, I mean, I think this is kind of, I don't know, it says genes and genomes. That's sort of obviously just a general statement there, but structure, function, interaction, and evolution, we're able to really study kind of all of this, having that information that we gained from sequencing or that human genome project. Okay. All right. Now, if we think about epigenetics, right, in epigenetic mechanisms, okay, we have to mention these because epigenetics, one, is probably a newer field. It's definitely a sort of hotly studied field. But the bottom line is that epigenetic changes impact disease or can cause disease. Okay, and so we're trying to identify what types of epigenetic changes can actually lead to disease, right? Can we actually use treatments to maybe reverse some of these. Okay, so important to understand this. So again, to kind of put things into perspective in terms of why a clinician would want to learn about these things. Why? Because it impacts disease. Okay. All right, so it says here, epigenetic mechanisms affect gene function. Yes, that's true. They affect gene function. They affect gene expression. There is no change in DNA sequence if we talk about an epigenetic change, right? If again, you want to kind of further clarify or define epigenetics, it's the study of changes in gene function that are heritable but do not change gene sequence. I guess that's the big thing, right? These changes are heritable, right? That's why we call it epigenetics, because they can be passed down. Okay. If you want to kind of take it now a sort of a larger scale and say epigenome, we're going to talk about chemical compounds that are not part of that genomic DNA that can then affect gene expression. Okay, so again, sort of larger, one step up, larger scale. Again, if you kind of think about this, so again, I mentioned this sort of epigenetic changes, right? These are changes that can be passed down, changes that affect gene expression. You can think of it on like the single cellular level. If we think about kind of like the development of a tumor or development of cancer, where you can have changes that are passed from one cell, from one cell to another during cell division, that then can lead to formation of a tumor or something like that. You can also think of it as these being passed down from mother to child, right? Or father to child. Okay, that they're inherited that way as well. Okay. Says here, we're mentioning epigenomic plasticity. Okay, so it says allows genetic reprogramming due to various factors. So meaning, epigenetic factors can be modified by lifestyle changes, things like nutrition, things like air pollution, things like chemical exposure, right? Just the aging process in general. All of these things will affect or can affect epigenetic factors. And then if those epigenetic factors are affected, it then can affect the function of genes, right? Okay. This is because if you kind of think about this a little bit differently too, like nutrition is not going to cause changes in DNA sequence, right? But can cause changes in epigenetic factors that then can affect how those genes function or are expressed. Okay, so I think that's kind of a nice comparison to think about. There are particular periods of sort of the life cycle that are particularly vulnerable to epigenetic changes, right? It says here deregulations, right? So if epigenetic factors are not regulated properly, okay, it can lead to, again, changes in gene function and gene expression, okay? So particularly during gestation, during neonatal, and also during puberty, and also during old age. These are particularly periods of time where your body would be, or your cells would be, highly vulnerable to changes. Again, if you think about gestation, well, here, this is a period of initial development where you have lots of cell division going on, cell differentiation, and so any slight change in a factor might really affect gene expression that would have a sort of a more detrimental effect, okay, on the body. So here are some roles. And so I kind of just mentioned this, right? It says essential roles of epigenetic modification. So it says cell differentiation, right? So again, if you think about during development, during gestation, we're talking about, a a fetus forming from just one single cell. Okay, so cells are produced, right? Your body, from that one single cell, you're going to have lots of cell division to make more cells, and then those cells need to differentiate into specific cell types. Okay, and so epigenetic factors are going to regulate or drive that process. And so if you have some sort of deregulation or problem with one of those factors, that's going to affect differentiation. It's greatly going to affect then development of that embryo and then the fetus. Okay. Also, there are, because we kind of mentioned right back here that, things in life like nutrition, right? Air pollution can actually affect epigenetic factors. Here's an example here that there are, food chemicals that can actually cause methylation of DNA. Okay, and so that's something that actually is considered an epigenetic process. And so if a part of the DNA becomes methylated that shouldn't, it can actually silence a gene, and actually so then that protein wouldn't be produced. Okay, medications can do things like this as well. And so again, I alluded already to this sort of DNA methylation. Also, histones become methylated, and that alters the activity of them. We said histones are the proteins that help package the DNA. Okay, so there are factors that can affect that as well. Also involved in chromatin remodeling, which is another, again, these are things that are beyond the scope of this class, but these are some specific biochemical processes that are controlled by, that we consider epigenetic. Okay, RNA interference. These are molecules that, again, can bind to RNA and, again, silence or affect the activity of the RNA, okay, or of the gene. All right, and so if you look here, this is a figure that's kind of showing you some of the things that I talked about here. The this down here is basically the figure caption that's in the book here. Okay. So it's showing you here epigenetic mechanisms, right? Affected by, remember we mentioned some of the things in the last couple slides that can affect these epigenetic mechanisms. It's showing you some examples of them, which were also listed before, right? So this is showing you here's a methyl group that's added onto the DNA, right? We call that DNA methylation. And again, if this methyl group is added on here, then this part of the DNA sort of becomes unavailable, okay? And so you can see that if these methyl groups are added where they're not supposed to be, it can lead to disease or some sort of issue, okay? So this is what this is showing you here, this sort of, right? It's inaccessible or it's inactive, okay? Yeah. So that's what this is showing you, right? So then again, it's showing you here's another epigenetic factor sort of binding here that then again causes this part to become inactive because it's causing the DNA to wind up around these histones. Okay, the bottom line is these epigenetic or anything that might alter an epigenetic factor is going to affect gene expression, meaning is that gene able to be transcribed and then translated into a protein? If it's supposed to be, say it's supposed to make a specific hormone, right? I don't know. Let's think of it's supposed to make insulin, right? But for some reason, it's silencing that gene. Well, then those cells aren't going to produce insulin. Okay, so hopefully that sort of helps a little bit as well. So then this is showing you that, yeah, any deregulation in these epigenetic mechanisms can lead to disease, right? So I'm mentioning some here, but really there are a lot of diseases that it can lead to. Okay, explain the function and sequence of events involved in gene replication, right? Mitosis, meiosis, and then transcription and translation. So I've sort of mentioned some of these terms along the way, so we talk about them a little bit more. Okay, so if we talk about replication is just going to be DNA replication is basically the cell is making another copy of the DNA, okay? And this is going to happen before a cell is going to divide, before mitosis, okay? And so that's replication. If we talk about transcription, and we're going to talk more specifically in a second, but if we talk about transcription, we're talking about going from RNA. We're talking about going from DNA to RNA. If we talk about translation, we're talking about going from RNA now to protein. Okay, so I haven't really mentioned RNA, and which is why it's listed here. So it says RNA is involved in protein synthesis. Okay, it says it's integral to creating tissues and organs. Yeah, you need that RNA intermediate to make proteins. I told you the proteins are the functional molecules of the cells. Okay, and it is formed from DNA. Like I told you, if we talk about transcription, we're going from DNA to RNA. Okay, it does have a different structure than DNA. It's called ribonucleic acid. The sugar is ribose instead of deoxyribose. And it also contains uracil instead of thymine. Okay, it's single stranded. It doesn't have that double helix structure, but in general has a similar structure. Okay, all right. So we're getting into here now to talk about DNA replication and cell division. Okay, so when we think about cell division, we probably think of the process of mitosis and meiosis. Okay. So again, if we talk about body cells, right, they all contain 46 chromosomes. Okay, we gave the exceptions before, so we just kind of put that in as a caveat. If we talk about sex cells, egg and sperm cells, they must contain 23 chromosomes. So the way your body is going to create these cells is going to be slightly different. Okay, so when we talk about, again, these two processes of sort of making more cells, right, the body's going to use mitosis. Okay, so process of cell division. And in mitosis, you will form identical copies of a cell. Okay, so like I told you, the DNA gets replicated, right? And then those cells divide. And so then you have two exact copies of those cells. If we talk about meiosis, it is also a process of cell division. However, that's not true. It does not form identical copies of gonadal cells. For sure, does not form identical copies. But it's going to produce the sex cells, the egg and the sperm cells. Okay. So if we look at, again, I just want you guys to know overview. I'm not going to, we're not going to learn about the phases of mitosis or anything like that. But we want to know that the purpose of mitosis, right down here says to take diploid cells, those that have two pairs of 23, that's what diploid means, and make two identical clones, right? So this is showing you here. Here's your cell. The body's going to replicate its DNA and then divide in two, and now you're going to have exact copies of the DNA. Okay, and then this is showing you the main phases of mitosis. Again, this is something you go over in basic biology. You probably went over in your anatomy and physiology class as well. We're not learning about the different phases of mitosis here. Okay, just as sort of just review and a reminder. Okay, it says this process is used to make all somatic cells of the body. Okay, so identical genetic content of somatic cells, non-sex cells. Okay. Also, it mentions here, and I think this is kind of, again, important to think about. So if we think about early on in development, I mentioned this before, we're starting with one single cell that got fertilized, right? A whole bunch of other cells need to be produced via mitosis, right? Identical cells, okay? So this process is going to happen a lot then. But then those cells will then differentiate, right? And basically they'll differentiate by turning different genes on and off to create, say, a heart cell versus a brain cell, right? Versus a skin cell, right? But the early on in development, when we talk about cell division, this is mitosis that's occurring. All right. If you think about like wound healing and things like that, you're going to have new cells that are produced, right? Via mitosis. All right. Now, if we talk about meiosis, and sorry, these words are so small, I probably can make them a little bit bigger. Let's see. Let's fix that right now, I guess. Huh? I don't know why they're so small. Whoa, that's even smaller now. Sorry about that. Let's make them bigger. That's a little too big. All right, that's probably a little bit better. Okay, so let me go back to that show. All right. So this is now showing you meiosis. So we said, so meiosis is the production of egg and sperm cells. So again, you can start with just a regular body cell, somatic cell, but now you want to produce egg cells or sperm cells. Okay. Now, again, we're not going through these details. Okay. And in fact, the process of oogenesis, the production of the egg cell, is a little more complicated than the production of the sperm cells. But the bottom line is here, you're starting with a diploid cell. Okay, but you're ending up with haploid cells. So this cell has 46 chromosomes. These are going to only have 23. Okay, and so that's what this is telling you here. Okay. I kind of just mentioned all of this. All right, again, we don't need to know the steps of meiosis. We don't need to know the details. We probably should know that oogenesis is the process that produces egg cells. Spermatogenesis is the process that produces sperm cells, okay? And by no means are they identical, okay? None of these cells are going to be identical to any of them, but you're getting essentially, you're going from diploid to haploid. And so actually, let me fix that because that's not true. Okay, it does not form. It forms. Sorry. They are all genetically different. So that's one thing about, and again, we're not going to even get into that, but they are by no means genetically identical gonadal cells. Okay, so let's just fix that right now. All right, I missed that before. Sorry. Okay, so the next thing we're going to talk about here is transcription. Okay, so again, what's transcription? We're going from DNA now to RNA because what's the ultimate goal here? It's to be able to produce a protein. Remember, I told you the proteins are the functional molecules of the cell. Okay, so the process, so we're starting with DNA. We have to go through transcription to make RNA, and then from there we can go through translation to make protein. Okay, so it's saying here, transcription is the process of reading a gene. I guess that's a kind of nice way of putting it. The idea is that, again, the cell wants to take this one specific gene and make a protein, right? What does it have to do first? It has to go through transcription of that gene, okay? All right. So it says here, an RNA strand is formed from a DNA template strand. So again, from the DNA, you're making RNA, all right? There are different types of RNA. When we're talking about transcription and sort of, well, I mean, I guess all of these, let's just go with that. So all of these types of RNA are involved in transcription and translation, okay? If we talk about messenger RNA, that's the RNA that's going to be produced from transcription here, okay? So it serves as that template for a protein. Transfer RNA is involved in translation, and it actually carries amino acids to that mRNA, okay, essentially. So essentially what that transfer RNA is doing is it's reading the mRNA, the information in the mRNA, and it's then matching it up with a corresponding amino acid. Remember, proteins are made up of amino acids, and so that's how then the protein is made, okay? Ribosomal RNA, translation is going to occur on the ribosomes, so that's part of the ribosomes. Again, I'm probably telling you more detail than I really am requiring you guys to know. I really just want you to know transcription is going from DNA to RNA, okay? Translation is going from RNA then to protein, okay? So if we look at a picture here, again, there's more details than you need to know, but it's nice to kind of see it in a picture form here. So here we're inside the nucleus. Here's your DNA. What's happening is the cell's making mRNA from DNA, right? So that's transcription. So this is transcription. That mRNA that was made from transcription leaves the nucleus, right? And then it goes to a ribosome where now translation is happening. So again, when we think about translation, we're translating this information that's in the form of this base sequence in the mRNA into amino acids, or which is eventually this is going to become a protein, okay? So hopefully that makes sense. Again, I want you guys to understand big picture. I'm not going to ask you about like, what's the specific function of tRNA or anything like that, but you can see that here. Remember, I just mentioned on the last slide, tRNA reads this information in the mRNA and then basically translates it into an amino acid, brings that corresponding amino acid. This is going to be your protein here that's made, okay? So this is showing you translation here. This is transcription, the making of the mRNA from the DNA. This is translation. All right, so again, if we kind of go over this, it's showing you this picture here. Here's your DNA, right? This is showing you sort of base triplets in the DNA, just because that's going to match up to these codons in the mRNA. Now here's your mRNA, which was made by transcription. Here's your tRNA around that's going to read the mRNA. It reads it in these groups of 3. and then bring the this is a typo here. I forgot that this has this typo. These aren't all going to code for methionine. They're all going to code for different amino acids, which are listed down here. Okay, so like this coding for alanine, this coding for glycine. Okay, threonine. So again, now when these amino acids join together, right, they form a polypeptide, which will ultimately become a protein. Okay, so this zooms in a little bit more on this. Again, it's adding a little bit more information in here, showing you introns versus exons. Okay, so always after the mRNA is made, you have to actually take out the introns. So this is showing you the introns. So the introns are the non-coding portions of the mRNA. Where you see these greens here, the exons, that's what's going to actually code for the proteins, right? Like we showed here. All right. Okay, so again, I already was mentioning translation as we went along. So again, the end result is protein formation. Okay, I mentioned this as well. A codon, again, is a grouping of 3 nucleotides in the mRNA that corresponds to a specific amino acid. So if you think of, or you've heard the term like the genetic code, that's what it's referring to. These codons in the mRNA that code for a specific amino acid. Okay, again, some of this beyond the scope of what we need to know. Again, we don't need to know details of translation here. Again, this is zooming in now just specifically on translation, showing you the ribosome, showing you the mRNA, the tRNAs, and the polypeptide sequence being produced. Okay, I'm going to pause here and then I'll pick up with another lecture, another recorded lecture to get the rest of this chapter. So we'll continue on learning outcome 2.5 for the